Good morning, students. So, in the last class, we focused on regarding this pentose phosphate or phosphogluconate or HMP pathway. So, up to that oxidative phase we have covered, and we started with this non-oxidative phase of the reaction also. That ribulose 5-phosphate, which is formed, is going to be isomerized to form ribose 5-phosphate, and then epimerization reaction is also going to take place, which is going to result in the formation of xylulose 5-phosphate. So now let us move on to the next step of this non-oxidative phase of the reaction, that is a trans-ketolase reaction. So what do you mean by this trans-ketolase reaction? So what it is going to happen here with the help of this trans-ketolase enzyme. So the 2 carbon unit which is going to be transferred from the xylulose 5-phosphate to ribose 5-phosphate, right? The ribose 5-phosphate which is formed from ribulose 5-phosphate through isomerization reaction in the last step, which we have noticed. So the here what is going to happen? The 2 carbon unit from the xylulose 5-phosphate which is going to be transferred to the ribose 5-phosphate to form a 7 carbon sugar that is called a pseudoeptulose 7-phosphate, right? Pseudoeptulose 7-phosphate, and even 3 carbon compound, that is 3 carbon unit compound, that is glycerolidate 3-phosphate, right? So this glycerolidate 3-phosphate is an intermediate of glycolysis, right? Okay, so for this trans-ketolase reaction to carry out, we require a coenzyme or cofactor, that is thiamine pyrophosphate, that is TPP. It is also called as TPP. So it is going to serve as a transient carrier of the 2 carbon unit in this particular reaction. So it is mainly involved in the transfer of this 2 carbon unit from this xylulose 5-phosphate to the ribose 5-phosphate. So in this particular trans-ketolase reaction, this thiamine pyrophosphate, which acts as a coenzyme or cofactor, which plays a very important role in this transient and acts as a transient carrier of this particular 2 carbon unit. So this reaction you can see here, xylulose 5-phosphate, as I told you, in the third carbon atom COH will be there, and which will be, in case of ribose 5-phosphate, you can make out this is an aldose, and xylulose 5-phosphate is a ketose. So here CHOH, CHOH, CHOH will be there in the 3 from second, third, fourth carbon of this ribose 5-phosphate. So the 2 carbon unit from the xylulose 5-phosphate, 2 carbon fragment, which is going to be transferred to either this ribose 5-phosphate to form a this glycerolidate 3-phosphate, and one more xylulose 5-phosphate from the 2 carbon unit transferred to ribose 5-phosphate to form pseudoeptulose 7-phosphate, that is a ketose sugar again, and glycerolidate 3-phosphate, that is again a aldose sugar. You can make out. So here also 10 carbon unit, again exactly the same unit will be maintained, that is 3 carbon glycerolidate 3-phosphate and 7 carbon pseudoeptulose 7-phosphate. So you can make out here, right? So this particular trans-ketolase, which mainly involved in the transfer of this 2 carbon fragment from the xylulose 5-phosphate to either ribose 5-phosphate or even to erythrose 4-phosphate also, that we will see in the next after trans-aldolase reaction. Again one more reaction will come, that is trans-ketolase reaction again. This particular reaction which is going to happen, the same mechanism which we have noticed in case of TCA cycle or the Krebs cycle, right? So there also pyruvate dehydrogenase multi-enzyme complex, which was also utilizing TPP as a prosthetic group or the cofactor or the coenzyme, which is involved in the particular mechanism, right? That is a 3, which involves 3 isoenzymes and 5 cofactors there, right? TPP, NAD plus FAD and all. So the same mechanism which we can notice here also, right? So coming for the non-oxidative phase of the 6th enzymatic reaction, so that is again trans-aldolase reaction. What is going to happen in trans-aldolase reaction? So here in trans-aldolase, it mainly focused on the transfer of a 3 carbon fragment. So it brings you about the transfer of 3 carbon fragment from the pseudoeptulose 7-phosphate to glycerolidate 3-phosphate to give fructose 6-phosphate. So you can make out only 3 carbon unit is transferred from pseudoeptulose 7-phosphate. So remaining will be 4 carbon unit, that is erythrose 4-phosphate. The formed product 4 carbon will be erythrose 4-phosphate, and 3 carbon which is transferred from pseudoeptulose 7-phosphate to glycerolidate 3-phosphate to give fructose 6-phosphate. Now it is going to be 6 carbon sugar, that is fructose 6-phosphate. Again fructose 6-phosphate is a ketose sugar, and 4 carbon Erythrose 4-phosphate is a aldose sugar. So you can make out in this particular slide. So how this 3 carbon unit which is going to be transferred to glycerol 3-phosphate to form fructose 6-phosphate. So you can make out, you can differentiate between this 3 carbon from the glycerol 3-phosphate and 3 carbon from the pseudoeptulose. So you can make out in the fructose 6-phosphate what are all the changes which has happened, right? So erythrose 4-phosphate is left behind that particular 4 carbon unit in the pseudoeptulose 7-phosphate, which you can notice in the erythrose 4-phosphate sugar, right? Erythrose 4-phosphate is again a aldose sugar. So this trans aldolase which mainly involved in the particular transfer of the aldehyde group, right? So which we have seen in this particular reaction. So what it is resulting in the formation of fructose 6-phosphate and 4 carbon sugar, that is erythrose. Again here also maintenance of 10 carbon unit sugar, whatever the pseudoeptulose 7 carbon plus 3 glycerol 3-phosphate 3 carbon, which gives you again 4 carbon plus 6 carbon. So again 10 carbon unit. So coming for the next non-oxidative phase of the 7th reaction, that is second trans-ketolase reaction, which we are going to notice here. So in this reaction, what is going to happen? A 2 carbon unit which is going to be transferred from this xylulose 5-phosphate to the erythrose 4-phosphate. Understanding? So again 2 carbon unit from the xylulose 5-phosphate is going to be transferred to the erythrose 4-phosphate, which was formed in transaldolase of 6th non-oxidative phase of the reaction to form fructose 6-phosphate, that is again 6 carbon unit. 2 carbon from xylulose to erythrose 4-phosphate, that is 6 altogether, to form fructose 6-phosphate and glycerol 3-phosphate, that is 3 carbon unit. So this fructose 6-phosphate and glycerol 3-phosphate are going to be directly enter into the glycolysis, because those are the intermediates of glycolytic pathway, right? Fructose 6-phosphate and glycerol. So altogether you have seen in this 2 molecules of fructose 6-phosphate and 2 molecules of glycerol 3-phosphate. So those are going to enter into the glycolysis. So you can make out in this slide very proper schematic representation or the particular diagrammatic representation with the structure. You can make out how the 2 carbon unit which is transferred from xylulose 5-phosphate to the erythrose 4-phosphate also you can make out in the last trans-ketolase, and in the trans-aldolase also how the 3 carbon unit which is from the pseudoeptulose transferred to glycerol 3-phosphate to form fructose 6-phosphate also you can make out, and how the 2 carbon unit from the xylulose 5-phosphate is transferred to the which is 2 carbon unit which is transferred to the particular resulting in the formation of your pseudoeptulose 7-phosphate and one more product what we have seen in the transaldolase reaction, right? So these are oxidative phase of the reaction, 3 enzymatic reactions, and these are the non-oxidative phase of the reaction which are reversible, whereas oxidative phase of the reactions are irreversible. So you can make out in this slide, go through properly, and you can understand very easily through this diagrammatic representation. So you can see the flow of the 15 carbon atoms from the pentose phosphate pathway, by which 5 carbon sugar that is converted to 3 carbon and 6 carbon, that is glycerol 3-phosphate plus 6 carbon, that is fructose 6-phosphate, and you can also make out here glycerol 3-phosphate and pseudoeptulose 7-phosphate, right? And then that is trans ketolase. TK is referred to trans ketolase reaction, IS is referred to isomerase, CP is referred to epimerase enzyme, and fructose 6-phosphate erythrose 4-phosphate is condensing to form, here you can make out trans aldolase reaction, how it is going to happen, resulting in the formation of this product, and through trans ketolase again fructose 6-phosphate and glycerol 3-phosphate, one more molecule is going to be formed. So altogether you are going to observe here 2 molecules of fructose 6-phosphate and 2 molecules of glycerol 3-phosphate, right? So this last formed glycerol 3-phosphate and fructose 6-phosphate entering into the glycolytic pathway. So you can see this balance sheet in order to understand very clearly this flow chart of this pentose phosphate pathway reactions. 5 carbon sugar, so C3, that is not referred to C5 is referred to ribose 5-phosphate plus C5 is referred to xylulose 5-phosphate. Trans ketolase reaction you will get glycerol 3-phosphate and pseudoeptulose 7-phosphate C7. C3 is again referred to glycerol 3-phosphate plus C7 is referred to pseudoeptulose, where the 3 unit is transferred from pseudoeptulose 7-phosphate to glycerol to form C6, that is fructose 6-phosphate, C4 erythrose 4-phosphate, transaldolase, again trans ketolase reaction, C5 unit, that is xylulose 5-phosphate, 2 carbon unit is transferred to C4, that is erythrose 4-phosphate to fructose 6-phosphate, to form fructose 6-phosphate plus glycerol 3-phosphate through transketolase reaction. So you can make out altogether 3C5, that is 3 carbon unit, 3 molecules of 5 carbon unit gives rise to 2 molecules of 6 carbon unit, that is fructose 6-phosphate plus 1 molecule of glycerol 3-phosphate. Overall reaction of pentose phosphate in the non-oxidative phase. You can see here this glucose 6-phosphate may be regenerated from either the glycerol 3-phosphate or the fructose 6-phosphate by the enzymes of gluconeogenesis. What you can see in the gluconeogenesis process, the synthesis of glucose, what we have already discussed. So coming for the this step, what we have seen in case of this 8th reaction, 2 molecules of glycerol 3-phosphate which are going to be condensed to form fructose 6-phosphate, then converted to glucose 6-phosphate. This step, which is going to be reversal of step 2 of the glycolysis pathway, what we are going to observe in the 8th step of the non-oxidative phase of the pentose phosphate pathway. This is one more flow chart, just a flow chart how this glucose 6-phosphate is undergoing enzymatic reaction till the last your glycerol 3-phosphate and fructose 6-phosphate through transketolase reaction, which is going to happen. This is all green color, which is representing you the oxidative phase of the HMP pathway. This yellow color is representing you the non-oxidative phase of the HMP. So you can understand very easily by going through this slide. And coming for the next topic, that is regulation of HMP pathway. So this regulatory enzymes, what we have come across in HMP shunt or HMP pathway, the regulatory enzymes are glucose 6-phosphate dehydrogenase and 6-phosphogluconate dehydrogenase. So both are involved in the synthesis of NADPH molecule, right? So both the reactions in the oxidative phase of HMP, we have seen these enzymes are involved in the synthesis of synthesis of NADPH molecule, right? So this both synthesis of both the enzymes is mainly induced by the insulin. Insulin is going to act as a induction molecule, that is it is going to induce the synthesis of both the enzymes, that is glucose 6-phosphate dehydrogenase, or stimulating it. It also stimulates the synthesis of this 6-phosphogluconate dehydrogenase. So during the well-fed state condition, I have already discussed while discussing the glycolytic pathway enzymes and even regulation of this gluconeogenesis and glycolysis, gluconeogenesis through the hormonal level, that is insulin and glucagon, what we have discussed. So in the well-fed state condition, your carbohydrate level after the meal will be very high. So the intercellular concentration, which is going to be enhanced, and this leads to the further enhanced oxidation of the glucose molecule through this particular HMP pathway or HMP shunt. So this particular regulation of this what is that glucose 6-phosphate dehydrogenase enzyme and 6-phosphogluconate dehydrogenase. So we can see how this entry of this glucose 6-phosphate into the pentose phosphate pathway is mainly sorry is mainly controlled by the cellular concentration of the NADPH. So what we have seen in this those two enzymatic reaction synthesis, we saw the synthesis of NADPH, right? So this can be controlled by the mainly by controlling the concentration of the NADPH, cellular concentration of this NADPH molecule. So this NADPH how we can control. So if the glucose 6-phosphate is entering, if the cellular concentration of this NADPH is so NADP plus is molecule is more, it is involved in the synthesis of this particular glucose 6-phosphogluconolactone by utilizing this particular cofactor or the coenzyme. So this NADPH, more production of this NADPH synthesis or the synthesis of more NADPH, which will act as a strong inhibitor molecule of the glucose 6-phosphate dehydrogenase enzyme itself. So it is acting as a end product, right? So it is acting as a it is acting as a inhibitor or feedback inhibitor of this particular enzyme, that is key regulatory enzyme, that is glucose 6-phosphate dehydrogenase. So more amount of NADPH production will act as a strong inhibitor of this glucose 6-phosphate dehydrogenase, which will be carried out in the first oxidative phase of this reaction involved in the synthesis of 6-phosphogluconolactone. So This non-oxidative phase can also be controlled mainly by the requirement of the availability of the pentose molecules. So the synthesis of this 6-phosphate dehydrogenase glucose 6-phosphate dehydrogenase enzyme is mainly induced by the increased level of the insulin after a high carbohydrate meal. So if after the high carbohydrate meal, so the insulin hormone should work and it will be very actively involved in bringing down the glucose level in the blood to the normal. Right, high level of the glucose can be observed after a high carbohydrate meal. So of course this insulin hormone should it will work and it will involve in it mainly induces this particular synthesis of glucose 6-phosphate dehydrogenase activity to further involve in the conversion of this glucose 6-phosphate to further other metabolites in the through this HMP shunt. This how it can be controlled. So particular coming for the after the regulation of this enzyme. So what is the main significance of this HMP? Very importantly, how this how this HMP pathway is very importantly playing a very important role in the particular either in the pentosis molecules or either in the production of this NADPH or any other acting as a free radical scavenging mechanisms or erythrocyte maintenance of the erythrocyte membrane integrity. So we will see one by one. So what this particular HMP shunt is a very unique in generating two important products, as I said you earlier, that is NADPH molecule and the pentosis molecule. So if you come for the first importance of the pentose, this HMP shunt where the exoses are converted into pentose. Of course, we have seen the exose molecule is getting converted into pentose, that is ribose 5-phosphate and as well as xylulose 5-phosphate. So the most important being this ribose 5-phosphate are its derivatives, which are mainly useful in the synthesis of these nucleic acids such as RNA, DNA, and even it is involved in the synthesis many nucleotides, other nucleotides such as ATP, NAD+, and even coenzyme A. So these are all the nucleotides can be synthesized by utilizing this pentose molecules, that is ribose 5-phosphate. So coming for the next importance of this HMP is importance of the NADPH molecule, which plays a very important role in the HMP shunt. So where the demand for when will be the demand for the NADPH molecule? So in case of biosynthetic pathways, of course the NADPH which is synthesized in the degradative pathways, so we can make utilize in the synthesis of some biosynthetic pathways where the NADPH molecule will be synthesized in case of amino acid biosynthetic pathways also, and fatty acid synthesis also, where fatty acid synthesis is going to happen in the liver, adipose tissues and in the mammary glands, which will be discussed by our Kokila ma'am. So even she is already discussing, right? And even cholesterol synthesis in case of the liver, and steroid hormone synthesis which can be observed in case of adrenal glands and ovaries, etc. So NADPH which will be used in the synthesis of certain other amino acids also, that is involved in the enzyme, especially in the enzyme example glutamate dehydrogenase activity. So glutamate dehydrogenase enzyme which will play a very important role in the conversion of glutamate to alpha ketoglutarate, that is glutamate to glutamine also. There also you can come across glutamate to glutamine synthesis, amino acid synthesis. So the enzyme main important role is mainly involved in the particular conversion of glutamate to glutamine is glutamine synthetase. In this case, glutamate to alpha ketoglutarate will be catalyzed by this glutamate dehydrogenase, where it is going to play a very important role there, the coenzyme, that is NADPH or the cofactor NADPH molecule. So this particular what is that importance of this NADPH which can happen in many enzymatic reactions, as we have seen. So now coming for the next free radical scavenging mechanism, how it plays a very important role. So this free radicals, especially you might have come across, that is superoxide and hydrogen peroxides. These are mainly these are continuously produced in all the cells, of course. So these are going to destroy DNA, proteins and as well as fatty acids and even other biomolecules, all biomolecules also, and in turn cells are going to be further destroyed, of course. So the free radicals which are going to be inactivated by the enzyme systems, especially containing the superoxide dismutase and as well as peroxide dismutase and even glutathione reductase enzyme. So this reduced glutathione, that is GSH, which are going to be regenerated with the help of this NADPH, of course. So the NADPH molecule is involved in the regeneration of this glutathione, and that is a GSH molecule from the oxidized form of the glutathione to reduced form of the glutathione, that is GSH. So you can see in this slide especially formation of this reactive intermediates from the molecular oxygen in the first line, where the oxygen molecule is getting converted to other reactive intermediate, that is superoxide, and further hydrogen peroxides H2O2 and hydroxy radicals, those are we can make very highly reactive intermediates and even water molecule, right. So these are the reactive intermediates which are formed from the molecular oxygen. In the B we can make out that is active form of the antioxidant enzymes, superoxide dismutase and other peroxide dismutase, how it is going to play a very important role in the conversion of these catalase enzymes and superoxides, dismutase, all these enzymes are involved in conversion of these reactive intermediates to the active form by the all these antioxidant enzymes in this particular slide. So next, coming for the how this erythrocyte membrane integrity can be maintained. So this NADPH, which is mainly required for the RBC cells, right, mainly by the RBC cells, in order to keep this glutathione, which will be in the reduced form, that is GSH, in the reduced state. So this reduced state, which will in turn will detoxify, it will help in the detoxification mechanism. What we have what you are going to observe, this detoxification which will be carried out by the liver enzymes, detoxification mechanisms by the cytochrome P450 monooxygenase especially. You can see all these mechanisms in physiology topics, what I think now you already studied in your first semester, or now you are going to study in the second semester. Just you might have understood this particular mechanisms, right. So by that particular detoxification, so this reduced form of the glutathione, which is going to detoxify the peroxides and even the free radicals which are going to be formed within the RBC cells, right. So this NADPH, glutathione, and as well as glutathione reductase enzyme all together, which is going to helpful in preserving this integrity of the RBC membrane or the membrane of the red blood cells, right. So this how it can maintain the erythrocyte membrane integrity by the utilization or the required amount of this NADPH molecule, which will help you in detoxification process, which will maintain the glutathione in the reduced state, which will help you in the detoxification of the peroxide and other free radicals in order to maintain the integrity of the RBC membrane. So what happens? The next question which we will arise here is, what happens if glucose 6-phosphate dehydrogenase is defective? So in that case, glucose 6-phosphate, main key regulatory enzyme of the HMP, is itself defective. So you can understand the thing, that is there will be no production of NADPH, that is insufficient, resulting in the insufficient production of NADPH molecule. So all the particular process will be stopped, that is what we have seen till now, that is the maintenance of the erythrocyte membrane integrity will be damaged. So damaging of the cells, RBC membrane is going to happen, and even this will leads to the further what is that NADPH molecule which is involved in the synthesis of other biosynthetic pathways, that and all will be stopped, right, which will translates into the insufficient glutathione, which will be in the which will not result in the formation of this reduced form of the glutathione. Further there will be all kinds of medical problem which is going to arise. So the glutathione is very important, and first of all, in order to maintain the reduced glutathione, so NADPH is very must and very important to maintain this reduced form of the glutathione. What is going to happen, this glutathione and erythrocytes, how this is going to play a very important role? See, GSH is very extremely important, especially in a highly oxidized environment of the red blood cell, right. So matured RBC cells which have no mitochondria, of course you all know, these are totally dependent on the NADPH molecule, that's why. So from the pentose phosphate pathway we can regenerate this GSH, the reduced form of the glutathione, from the oxidized form, that is GSSG, by glutathione reductase enzyme. So only by the availability of this NADPH molecule. So if you are going to synthesize more NADPH, so that in turn involve in the synthesis of this reduced form of the glutathione and conversion of this oxidized form of the glutathione to reduced form of the glutathione, regenerating back by the enzyme glutathione reductase can be maintained. So you can maintain even the RBC membrane integrity, erythrocyte membrane integrity of the RBC membrane, right. So in fact, you might have come across, I have already discussed in the last class while discussing the introduction, about 10% of the glucose consumption by the erythrocyte is mainly done by this pentose phosphate pathway. So of course we require this NADPH molecule in order to maintain the erythrocyte membrane integrity, and as well as reduced form of the glutathione, by making availability of the NADPH molecule in order to carry out this particular process or the mechanism. So this reduced glutathione also detoxifies peroxides, what we have seen in the last slides, and where the cell with a low level of this reduced glutathione are mainly susceptible for the hemolysis to happen, right? Breaking down of the RBCs, that is hemolysis is going to happen. So individuals with a reduced glutathione are mainly subjected to the hemolysis. This is often clinically, which we can observe in the formation of black urine under these conditions, certain conditions of this hemolysis. So this glucose 6-phosphate, you make it as not D, that is glucose 6-phosphate dehydrogenase activity impairs of the cell to form the NADPH, which is mainly essential for the maintenance of the reduced form of the glutathione, which results further in a decrease in the cellular detoxification of these free radicals as well as peroxides which are formed within the cell. You can focus that particular process, how it is happening in this slide. So maintenance of the erythrocyte membrane integrity, how this reduced form of the glutathione, which will be maintained by the cellular activity of this NADPH, and if that is deficient, how this activity impairs the cell, which will result in the formation of NADPH, which is mainly required for the maintenance of GSH, and which will further lead to the decrease in the cellular detoxification of this free radical mechanism and peroxide formation. What is going to happen within the cell further leads to the cell damage, of course, right? So this prevention of this methemoglobinemia can also be maintained or done mainly by the reduced form of the glutathione. So it is very essential for the normal RBC structure maintenance, and by keeping this hemoglobin in the Fe2 plus state, right? So Fe2 plus state is a very important state in order to maintain this RBC, normal structure RBC. So this GSH plays a very important role. So this NADPH is also required in order to keep this iron of the hemoglobin, which is in the reduced state, that is a ferrous state, and to prevent the accumulation of this methemoglobin formation. This methemoglobin is nothing but which will lose the capacity to carry the oxygen molecule. So this glucose 6-phosphate dehydrogenase deficient persons, which will show increased methemoglobin in circulation because of the low level production of the NADPH, which further involve in the deficient production of the particular which will fail to maintain this GSH in the reduced state, which is essential mainly for the maintenance of the normal RBC structure, and by thereby keeping the hemoglobin in the Fe2 plus state. So all these is going to be reduced. So it further leads to the methemoglobinemia, and methemoglobin is going to be observed in the person who is deficient of this glucose 6-phosphate dehydrogenase enzyme activity in case of the blood. So coming for this detoxification of the drugs, last topic of this particular significance of HMP. So most of the drugs which are, as usual, you all know, other foreign chemicals or the foreign substances which are considered as xenobiotic compounds, which are going to be detoxified by the liver microsomal enzyme, that is called as CP, cytochrome P450 monooxygenase enzyme, with the help of this NADPH. See, here also this NADPH molecule is playing a very important role in the detoxification of this particular foreign substances or the xenobiotic compounds. So this detoxification of the hydrogen peroxide can also be inhibited, and cellular damage, which is going to further result through this lipid peroxidation, which further leads to the erythrocyte membrane breakdown and further leads to the hemolytic anemia. What we have seen, the hemolysis, because of the not because of the non-availability of this NADPH molecule. So most of the glucose 6-phosphate dehydrogenase deficient individuals are asymptomatic. They are only in combination with certain environmental factors which can be observed, that is sulfa drugs, and as well as antibiotics, and even aspirin, as well as antimalarial drug. So these particular factors which will be observed in combination with this particular deficient persons, so they are going to show that they will not show any symptoms. That is asymptomatic condition which we can observe with this combination with the environmental factors. So you can also see, in case of the lens of the eye, maximum concentration of this NADPH can be seen in the lens of the eye with this particular deficient person. And this NADPH is also mainly required for preserving the transparency of the cell lens. That's why how this NADPH molecule is overall involved in the maintenance of this total metabolic activity in our body. Very importantly, it is playing a role, first of all, that is the maintenance of the reduced form of the glutathione and maintenance of the erythrocyte membrane integrity, and as well as maintenance of the preserving the transparency of the lens, because with the production of this NADPH molecule through this glucose 6 phosphate dehydrogenase enzyme activity. If this is deficient, so of course all other mechanisms which will be stopped, other detoxification mechanisms as well as particular peroxidation mechanisms in order to maintain the erythrocyte membrane integrity, all will be stopped. So this further leads to the medical problems. So thank you. So in the next class we will further focus on the next other metabolic pathways. If you have any doubts, you can ask me, or please go through all these slides and be prepared. I may give you the test in the next coming week. Thank you.